we'll make a start. So today we have Professor Cam Simmons talking to us, and Cam is yet another graduate of the department. He did honours and PhD here and then postdoc in the UK for a while, and then got into more of a global health sort of perspective and lived in Vietnam for quite a number of years. And now he flies around the place still looking at trying to help people in developing countries and other areas, and he has a strong interest in dengue, so some of you may have heard him talk about dengue in second year last year. Okay, and so he's going to talk more about the global health side of vaccination, vaccine use and so on today. And you need to make allowances. He flew in from Indonesia at 7:30 this morning and has brought a lurgy with him, so be kind.
Thank you, Louise. Good morning. So I'm hoping my voice holds out, so forgive me if I crack up or cough, etc. So we're going to talk in the first half of the lectures, talking about a very practical tool that we use to fast-track vaccine development, to make new vaccines for diseases that we don't already have vaccines for, and also just a fundamental underpinning to understand how the immune system works to express immunity, whether that be vaccine-elicited immunity or immunity elicited after a natural infection. So we're going to start just by talking about why are correlates of immunity. And here we're talking about correlates of immunity to infectious diseases. And that immunity might be elicited by natural infection. For example, a Salmonella typhi, a typhoid infection, is generally an immunising infection, but so is immunisation with the VI antigen from Salmonella typhi. So there's different ways of eliciting immunity to infectious diseases, and correspondingly different correlates of immunity. So why are correlates of immunity important? So it tells us about how the immune system works. It tells us about mechanisms of immunity. Correlates of immunity are important because it helps us to refine vaccine candidates. In the old days, if we wanted to make a vaccine against pathogen A, the first thing we do is probably just grow it up and try and kill it, and then stick it in someone's arm. Well, we can be a bit smarter now with molecular biology and making recombinant subunit vaccines. We've got the possibility of making quite clean, quite clever, rationally designed vaccines, but it needs some understanding of how the immune system mediates immunity. Correlates of immunity are important when you're making a second generation vaccine from an existing one. So, it's also a critical aspect of manufacturing, day-to-day manufacturing of vaccines, that the influenza vaccine that you may or may not have received at the start of this flu season, every season it gets tested for potency with the immunisation of animal experiment, and showing that antibodies raised to the vaccine neutralise the viruses in the vaccine. It doesn't work very well when the influenza strain circulating in the population changes as it has done this year. Correlates of immunity are important because it helps population-level vaccine policy decisions. If you've got an immunological marker that reflects immunity, and you can readily measure that in a population, you can understand the need for vaccination in that population, and you can understand what's called the vaccination threshold. What proportion of the population, what percentage of the population do you need to immunise to reduce what we call the epidemiological reproductive number, called R0, to below 1. When R0 drops below 1, transmission of a particular infectious disease stops. Immune correlates are really important when you can't do a phase 3 efficacy trial because it's unethical, or because the disease that you're studying is relatively rare and it would just be practically really hard to do such a trial. So as an example, meningococcal disease, these vaccines are not registered, licensed on the basis of large phase 3 trials with hundreds of cases in them. They're registered on the basis of immunogenicity, safety data, and laboratory work that shows that antibodies to the meningococcal bactericidal. And then, as I mentioned before, bridging from first generation to second generation vaccines. Some of our influenza vaccines, we're making them the same way we've been making them for 70 years, growing them in eggs. But in influenza, there is an accepted immune correlate of immunity, antibodies to the hemagglutinin. And so if you want to make second and third generation influenza vaccines, you use that understanding that a particular antibody threshold to the hemagglutinin protein is a correlate of immunity, and so that's how you guide your second and third generation vaccine generation. How do you identify correlates? Well, it's not easy, and so people look to different systems. One of the natural systems that exists is infants born to women. Those infants acquire the IgG population from mum, but they don't acquire any of their T cell response. And so you can look at infants at their risk to particular diseases according to their month of life. And so in the first month of life, the infant is full of mum's IgG repertoire to all of the infections that she's seen over a lifetime. And what you see generally is that infant's immune to multiple infectious diseases in the first few months of life. But then those antibody titers from mum wane in the baby over time, and they become susceptible to the infections around us. We can look at, in vaccine trials themselves, we can look at what is it in vaccine efficacy trials that people who were protected with the vaccine, what sort of immune response did they generate in response to vaccine that didn't happen in the control arm of the trial? What were the key features associated with vaccine-elicited protection? You can also try and understand in both natural infection but also in vaccine trials when people, for example, have received a vaccine but then subsequently get the disease. So it's really, I guess, a negative correlation. What is it about their immune response profile that led to them still being susceptible to a particular infection? And then human challenge studies, really growing as a scientific space and as a way of fast-tracking vaccines. And human challenge studies involve healthy participants, almost always adults, volunteering to receive a pathogen, whether it be influenza, whether it be dengue, whether it be respiratory syncytial virus. So by doing studies of healthy volunteers who you can extensively study at baseline before you experimentally inoculate them with a pathogen, you can seek to understand immune correlates of protection. You can also do animal studies. Mice are not humans, but increasingly we've seen examples in the vaccine space of non-human primate studies being used for licensure of new vaccines, because those animal studies demonstrate that a given vaccine is able to protect non-human primates from a particular pathogen, and that it's possible to identify a possible correlate of immunity in those animal studies. And we'll talk about the animal rule a bit later. So just some definitions. So what do we mean by a correlate of immunity? It's an immune response, and most of the time in the vaccine space, it's antibody responses. There's not a good example really of a licensed vaccine where a particular type of T cell response is a correlate of immunity. And a correlate can be mechanistic or non-mechanistic, and we'll unpack that a little bit later. But clearly it suggests that the immune response that you're measuring may be mechanistically involved in preventing you from getting an infection, or it might be a proxy, a correlate of something else that you haven't measured or you don't know how to measure that sits underneath that correlate. An absolute correlate is a specific level, a defined threshold, for example, of antibody that is highly correlated with immunity. And we have examples of this in hepatitis B, for example, where we understand that a certain number of units of antibody to the hepatitis B surface antigen is enough to offer protection from hepatitis B infection. So that's an example of an absolute correlate. A relative correlate is something that is on a continuous spectrum. And so, for example, against dengue viruses, for example, we have a strong sense that that there's not an absolute correlate for protection against the dengue 2 virus, but if you've got a lot of antibody to the dengue 2 virus, it's very likely you're immune, clinically immune to that infection. If you haven't got very much of that antibody, particularly to the envelope, you're more likely to be susceptible. But it's along a gradient of risk, so it's not an absolute threshold. It's not a precise number or titer of antibody. A relative correlate is a level of response that's variably correlated with protection, and we'll unpack a little bit of this with examples in a few slides later. Co-correlate is when you've got multiple actors contributing to the expression of immunity. And an example here is rotavirus vaccines, where in fact IgG and IgA antibodies may each represent correlates of clinical immunity. So a mechanistic correlate is an immune response that is responsible for protection. So if you've got this immune response, we're very confident that you're going to be clinically immune to that infection. A non-mechanistic correlate, an immune response that substitutes for the true immunological correlate, which may not be known. We don't know everything about how the expression of immunity works to pathogens. Tuberculosis, a great example. We have one lousy vaccine, the BCG vaccine against Mycobacterium tuberculosis. It's not a very good vaccine, and we don't even know how it works. We don't understand acquired immunity to tuberculosis well enough to come up with better vaccines. So, when we talk about correlates, there's a whole lot of precision involved in considering them. And so, first principle is that when you talk about a correlate of protection, what actually do you mean? Do you mean against infection? So any infection. So in other words, we're talking about sterilizing immunity. So there's no possible detection of the pathogen of interest in someone who's been vaccinated. Or are you talking about protection against disease? So that you might tolerate someone being infected, but as long as they don't get sick, you don't care. That's fine. And so in the vaccine world, many of our vaccines work through sterilizing immunity. Some of them we're pretty confident only protect against disease, but allow subclinical infections. And so there's a level of precision you need to bring into your thinking about correlates. And so, as an example of this principle, so protection against polio, for example, the correlate is IgG serum antibodies to the polio virus. That's protection against disease. They'll stop you getting poliomyelitis. They'll stop you getting sick with the polio virus. If you want good, robust sterilizing immunity so that you don't get infected at all, you need IgA and IgG antibodies. So that's an example of sort of co-correlates that are correlates against infection, outright infection, as opposed to protection against disease, where you might accept some subclinical infection. And again, in this precision, also in pneumococcal disease immune correlates, that pneumococcal disease is famous for having different expressions of disease, whether you're in the elderly age group or whether you're a young infant. Some of these are more common. Otitis media, really common. Inner ear, middle ear infection for children. If you want protection against otitis media, you need quite a bit more antibody than you need to protect against bacteremia, which is disseminated bacterial infection such that you find bacteria in your blood. If you want to prevent colonisation or nasopharyngeal carriage, you need even a bunch more antibody. So really ratcheting up the level of antibody that you need as a correlate to protect against these different I guess, expressions of colonisation or infection with the pneumococcus bacterium. Of course, correlates of immunity are only relative to to what you're getting challenged with. And so, we know that for example, in human challenge studies, one of the complexities is working out how much to give human volunteers of a particular pathogen. We'd like to think we know in the natural world, in the community, what the dose is you get of influenza, for example, when you're out in the community. But the reality is we don't. We don't have a great idea of, when you're naturally exposed to influenza in the community, how many virions did you cop to get your clinically apparent infection. And so correlates of immunity, again, have got this context of it's a bit dependent on what the challenge dose is. And so most of the time when we think about challenge doses, most of the time when we're thinking about correlates, it's in relation to the way vaccines perform in large efficacy studies, and so it's related to natural exposure. But increasingly, as human challenge studies come along, correlates have got this added complexity that it's related to how much pathogen you're putting into a particular host. And so here's an example with challenge of a polio vaccine by oral poliovirus. So here's using the live attenuated oral polio virus as actually a challenge strain. And what we're looking at here is protection after low dose challenge with the live, really using it as a challenge strain, protection after low dose challenge or high dose challenge. And you can see that whether these vaccinees received the oral polio vaccine or the inactivated polio vaccine, there's a difference whether you challenge them with a low dose or a high dose. So just a very practical example that's becoming quite common in human challenge studies now. And so when we think about correlates, we think about IgG, for example, IgG titers to a virus, for example. And it's not just about antibody binding to antigen. It's not a simple measure of just how much antibody is there and how much antigen can they bind. What we're interested in is generally our correlates of immunity are functional antibody responses. And so, here's an example. Here's an example with Group C meningococcal polysaccharide vaccine. These are ages of children who were immunised, some adults down here. This is after immunisation. This is the percentage of vaccinees who were ELISA positive, so had antibodies that bound the meningococcal polysaccharide on an ELISA plate, so very essentially a biochemical reaction. But this is the percentage of vaccinees who had functional antibody that could be lytic for the meningococcal through opsonization and functionally killing the meningococcus. And so the take-home message is correlates tend to be, most of the time when we measure them, they're not just biochemical measures of how much antibody to a given antigen, but some functional measure of that antibody population. And for viruses, typically it's about virus neutralization. And so whether that's for influenza, whether that's for dengue, whether that's for our EPI vaccines that we've all received as young children, this is about virus neutralizing antibody most of the time. So this fourth principle is that most current vaccines, and particularly our EPI vaccines, MMR, for example, most of these vaccines work by eliciting antibodies that are neutralizing for the virus. And to a large extent, that's because we've been able to do this because it's relatively easy. It's not sophisticated technology to measure virus-neutralizing antibodies in a serum specimen from a vaccine recipient, and to measure that virus neutralization against virus in a laboratory that has been added to a culture system. And so it's not new technology, it's old technology, but it works. New vaccines are going to have to be that perhaps where antibody is not a simple correlate or a mechanism of immunity, are going to be increasingly more complex. For example, against tuberculosis, against HIV. These are almost certainly going to need to be T-cell-based vaccines, and that's a more complex challenge to measure those responses. So these are examples of major licensed viral and bacterial vaccines for humans according to the mechanism of disease prevented by the vaccine. And so essentially all of these viral infections, mucosal replication for influenza rotavirus, human papillomavirus, prevention of retrograde transport of the rabies virus to the central nervous system, all antibody against bacteremia and against toxin production for these bacteria. Of course, your tetanus toxoid vaccine, your diphtheria toxoid vaccine that everyone's received as children and you will have been boosted at later points in your life, are all about making antibodies to prevent the function of toxins produced by these bacteria. So most of our arsenal of vaccines is really simply driving antibody responses to critical components of the vaccine to neutralise the pathogen or to neutralise the toxin from the pathogen. So let's get a bit more complex. Memory can be a correlate of immunity, a mechanistic correlate of immunity. So here's an example in the context of hepatitis B. And so here we have it's a large observational study in China, and we've had 50% of the vaccine group have got anti-surface antigen antibody. The controls have also, 1/3 of the population have also got anti-surface antigen antibody, and that's because in endemic settings, hepatitis B infects many people. Most of the time, it's a self-limiting infection, and people seroconvert to the surface antigen. When they looked 15 years after immunisation, only 2% of the immunised children were surface antigen positive, and this is a marker of chronic hepatitis B infection, versus nearly 17% of the controls. And so what was happening here, and so this was sort of the estimated efficacy over that 15 years, what we think happens in the context of hepatitis B is that when hepatitis B infects you, whether you've been immunised or not in the past, it's quite a slow replicating virus. So it's not a quick, dirty, acute infection like dengue or the influenza virus. It's a bit of a slow burn. And so if you've got, you may not have spectacular existing levels of antibody to hepatitis B surface antigen, but because the virus is a bit of a slow burn in its replication, if you've got some memory B cells that have been laid down from immunisation, they've got time to wake up, to be re-stimulated and start producing anti-surface antigen antibodies. And so hepatitis B is a particular example where we have good evidence that you may not have good antibody titers to the surface antigen, but we think very likely you're clinically immune because of the time window, the slow burn of hepatitis B infection, and that's just the natural history of hepatitis B. So examples, and you know, we don't have great examples for this, but T cells as memory, you know, there isn't a licensed vaccine for which T cells are a recognised correlate of immunity, but you can imagine if you wanted to effectively do therapeutic immunisation for HIV, you're going to need to lay down, you know, a broad profile of T cells to the HIV virus. We know that it's likely that for some T cell responses, for example, against dengue, those fast-acting, circulating effector memory T cells are most likely to be important to an acute viral infection like dengue, like influenza. But for other viruses like back to hepatitis B, it may be that your central memory population of T cells has got time to recognise this, I guess, slow-burning pathogen and respond. But the whole T cell space, again, to reiterate, There isn't a licensed vaccine for which T cells are a correlate of immunity. So, to reiterate, to conclude this bit, almost all current vaccines work through antibodies in serum or on mucosa. For example, an enormous amount of work to develop rotavirus vaccines that suggest that certainly to get sterile immunity against rotavirus, you need antibodies at the mucosa, at the gastrointestinal mucosa. And very often, from animal studies, we believe these to be mechanistic correlates of immunity. So they're the part of the immune system that's responsible for preventing that pathogen infecting the host. Functional characteristics, we've talked about that, most of the time we need to be measuring the functional attributes of antibody populations. Most of the antibodies you make to a vaccine antigen or to a natural infection, most of them are not functional. They'll bind antigen, but serve no great role in the expression of immunity. And so antibody might be highly correlated. So again, most of our vaccines we think antibody is actually a correlate of immunity, or it's synergistic with other functions. So it's a correlate, but not a mechanistic correlate. It's a marker of some other aspect of the immune response that protects you. And so I want to now flip to, you've heard in a previous lecture of how vaccines get made, and you've been taught, and as decades of immunologists and vaccinologists have been taught, that vaccines take a long time to develop. It's a slow empirical process that you need lots of safety data. You need to do stepwise phase 1, phase 2, phase 3 clinical trials. You need to spend somewhere between 500 million to 1.2 billion on average to deliver a vaccine to licensure. So everyone, what does this slide represent to you? What's this? What disease? Infectious disease? Ebola. Yeah, very good. So Ebola gave the world a wake-up call, a real kick up the backside to say we need to do things differently. And we know how to control Ebola. So, Central Africa's dealt with Ebola for decades. There's sensible measures to prevent Ebola transmission, and that's how Ebola's been shut down in the past. But clearly, the outbreak of Ebola in Sierra Leone, in Guinea, was a real potential global disaster that killed over 10,000 people. And so it was a real kick up the backside for the global health community that we needed to rethink our process of making vaccines for emerging disease threats. When the Ebola outbreak kicked off in 2015 in West Africa, there was no licensed vaccine for Ebola, but there were a number of candidates that had been tested in animal studies, small and non-human primate animal studies. And of course, you will have seen similar slides to this, but the world is clearly much, much smaller. So this kick up the pants was, not just about, it was a little bit of self-interest. Ebola gave us a self-interest kick up the pants in that it may not be Ebola today, but it might be the MERS coronavirus from the Middle East. It might be a funny coronavirus that appears out of Southeast Asia. It might be a novel influenza recombinant out of China. But the world is a much, much smaller place, and we know that these pathogens take a ride on airlines really, really quickly. And so there's been an enormous effort over the last 3 or so years to change our thinking and to change the mechanisms to make vaccines against emerging disease threats like Ebola, but also against others. And so this is a slide, you've probably seen something similar. This is the typical timescale for vaccine development, the steps involved: antigen identification, make the antigens, preclinical testing, maybe phase 1. Most vaccines never get beyond phase 1. In fact, most vaccines never get to a person. Most vaccines never get to phase 1 testing. Phase 2, about 40% of vaccines fail at phase 2. They don't go any further. Some vaccines carry on into phase 3 testing and then into licensure, and then in in a post-licensure world, you need to keep doing research on your vaccine. So this is a 9 to 14 year effort if we follow the traditional pathway. Just to flick back to Ebola and how Ebola has sort of changed the thinking. So I've already told you that candidate Ebola vaccines had already been trialled in animal studies. And part of this was driven because of Ebola's on the US bioterrorism list as pathogens of interest. So again, self-interest here. Another key coming together was, and this has been around for some time, the so-called animal rule that would allow accelerated development of vaccines or drugs based on results from animal studies that were acceptable to the field. And so I'll come back to the animal rule in the last slide. But the animal rule says that you can do a variety of that human studies and animal studies don't necessarily need to be distinct scientific enterprises, but they can run together in the development of a vaccine candidate. And so here you've got typical human studies on this side, phase 1 safety, phase 1 expanded safety. So this is more larger studies to understand the safety and immunogenicity of a vaccine. But here, the route that the Ebola vaccine candidates took was optimization of composition, dose timing, and immunogenicity in animal studies. Clearly, doing Ebola vaccine trials in people is all very well, but in the Western world, you're not going to understand about immunity. With non-human primate studies of Ebola vaccines, you can challenge those non-human primates with Ebola and understand clinical immunity from a challenge infection. And so what happened in 2014 and 15 when the Ebola outbreak kicked off in West Africa was a whole acceleration of the animal rule testing approach for candidate Ebola vaccines along this road. So a real enormous amount of energy to triage what were the best Ebola vaccine candidates and did they work in the best available animal challenge studies. And so at the back end of those animal studies was a WHO meeting in September 2014 and was the recommendation to pursue 2 candidate Ebola vaccines in clinical studies with a view to taking them to Africa. And I'm really only going to talk about the winner of this competition, and that was the recombinant vesicular stomatis virus, so a plant virus that's been engineered to express the glycoprotein from the Zaire Ebola virus strain. And so here it is, just unpacking that. So this is a traditional virus vector approach using, in this case, a plant virus to vector in. That you administer this as a live virus. It replicates in a very transient way, subcutaneously, and expresses the Ebola glycoprotein, and the immune response sees the Ebola glycoprotein, the major surface protein on the Ebola virus. So this is the preclinical history of this vaccine. This was shown to be immunogenic, so that when you immunise monkeys, they made antibodies to the Ebola glycoprotein. And animals that were then challenged with a lethal dose of Ebola virus were clinically immune, so they didn't die, they didn't get sick. And then there are a set of assay development to understand, well, if we understand that these macaque monkeys are immune from Ebola virus challenge, what is it about their immune response to this vaccine that makes them immune? So it's they're looking for an immune correlate. And so there's a whole lot of assay development to come up with what we thought was the best immune correlate, which is effectively virus neutralization of the Ebola virus and driven by anti-glycoprotein antibodies elicited by the vaccine. We don't really understand still yet what the minimum antibody titer is for immunity in humans, but we'll talk about the clinical work in a moment. And of course, what this needed was to launch from what were animal studies straight into the field in Africa. How never been done before. How could you fast track what would be normally a 5 to 7 year clinical development pathway, really careful stepwise testing, to go straight from animal studies straight to 3 of the poorest countries in the world who were affected by Ebola. And so we'll talk about first of the phase 1 study. This was about understanding how much of the vaccine to give, to understand how immunogenic it was under different doses, and to focus on severe adverse events. So severe adverse events are those clinically important events that occur in response to vaccination. And so this was the vesicular stomatis virus vector being used here was novel. It had never been used before, and certainly not with an Ebola glycoprotein in it. And so the results from the phase 1 safety studies, which were done variously in Western Europe and in healthy volunteers from other parts of Africa, not West Africa where Ebola was raging. Several phase 1 studies that were all done concurrently. And the bottom line really from those safety studies is that this was a very well-tolerated vaccine that showed no significant safety signals. And all of these phase 1 safety studies were squeezed into about a 2-month time window, run concurrently in different parts of Western Europe and in Africa. The really important thing was that all of the healthy volunteers in those phase 1 studies made Ebola glycoprotein specific antibodies following vaccination. In this case, for speed, these were measured by ELISA, but there were bridging studies to show that what you measured by ELISA correlated to a certain level with virus-neutralizing antibodies to the Ebola virus. Clearly, it's not easy to measure virus-neutralizing antibodies to the Ebola virus because you need a PC4 laboratory suite to do that work. And so the bottom line is that all of this was really promising to take this vaccine candidate forward. And so what they did was, in the middle of the outbreak, we're now 2015, was to do effectively an efficacy trial, to launch from animal studies, a small number of phase 1 safety immunogenicity studies, right into an efficacy trial, and really break all of the vaccine development rules. And the way this trial was done was clever. It's a ring vaccination trial, and what that means is that healthcare workers and together with the research team identified Ebola positive index cases, and then what they did was to randomise each index case, the contacts of each index case, to either the placebo or to the Ebola vaccine candidate. And so there were 90 index cases, so that's 90 individuals with Ebola, and therefore 90 clusters set up. And a cluster is the contacts of one index case. And so I said incorrectly said there was a placebo. There wasn't a placebo, and there was a big point of contention for the field. Is it ethical to do a placebo-controlled trial for Ebola? And so what the team decided to do was to compare early immunisation versus late immunisation with this Ebola candidate. And so there were 48 clusters and 2,000 contact participants who received immediate vaccination. So that's within a couple of days of the Ebola index case being identified, and then delayed vaccination. And this is a couple of weeks after the index case was identified. So we're comparing immediate immunisation with this candidate versus delayed immunisation with 2,000 and 2,000 roughly in each arm. And a really tough, this is the setting, really difficult cold chain, really difficult place to do a clinical study in a community ravaged by Ebola. And so the really good news is the vaccine was 100% efficacious in preventing Ebola disease in contacts of the index case. Effectiveness was 100% compared to the delayed vaccination arm. Okay? And so, and this is sort of borne out by this is a survival analysis of, so on this y-axis we have individuals with confirmed Ebola virus disease. So this is percentage of the cluster participants, so 2,000 in each arm. And this is the arm that had immediate vaccination. So you can see one or two, each step here, if you like, in the staircase, represents one new Ebola case in the vaccine recipients. And so you can see there's three Ebola cases in the immediate vaccination cohort, but there's a whole lot more in the delayed Ebola vaccination cohort. And so this is now really the standard of care. And so for the last Ebola outbreak that occurred earlier this year, there are stockpiles of this vaccine sitting in Africa and in Europe that can be delivered for ring-fence vaccination for communities where Ebola has started its transmission. There are lots of questions about how you use Ebola vaccines and who's going to pay for them. Entirely sensible to have stockpiles for ring-fence vaccination approaches in communities. Entirely sensible to think about having some high-risk groups, for example, healthcare workers, particularly frontline healthcare workers in settings where Ebola is historically known to pop up, to laboratory workers who might be diagnosing clinical samples that might have Ebola in it. What do you do for the general community? Is there an argument for immunising some of these these populations in Ebola-affected regions? Probably not. Probably not cost-effective. Ebola is a very sporadic, relatively uncommon disease, and this is still not a cheap vaccine. Certainly not a cheap vaccine to deliver to the places that need it. Is it possible to do post-exposure prophylaxis with this vaccine? So let's say you're a healthcare worker who's unknowingly come into contact with an Ebola case in a hospital setting. You might be a nurse or an emergency room doctor, and you came into contact with an Ebola case without knowing it. Can this vaccine work for post-exposure prophylaxis? And there's animal studies to try and address that at the moment. And then it's all very well to have a vaccine. How do you have it in quantities and logistically available so that it has a public health impact? And so, having ripped up the rule book a bit about how you make vaccines to emerging infectious disease threats, there's been a lot of other effort to try and create a framework so that companies and academic groups have got that there's an outcome possible for vaccines against emerging disease threats, whether that's the MERS coronavirus in the Middle East, whether it's the Lissa virus or the Marburg virus or the hantavirus. And so this is a strategy called CEPI. CEPI is a Coalition for Epidemic Preparedness Innovations. It's the coming together of funders from it's around 12 countries with leadership from industry and academic partnerships that is creating the possibility for vaccine companies and academics to make vaccines against emerging disease threats, and for those vaccines to get tested all the way into phase 1 safety and immunogenicity. So that you don't need, if the MERS coronavirus or a funny influenza out of China suddenly became transmissible, highly transmissible for humans and started spreading globally, which is entirely plausible, that we wouldn't be stuck at the animal or laboratory phase for making vaccines against these diseases, that we would have invested into the science to have the animal work completed and phase 1 safety and immunogenicity study done for vaccine candidates so that we can move much, much quicker to respond to these public health threats. And so this is the vision of CEPI, that it's really buying insurance. And so what it's doing at the moment is focusing on the lyssavirus, on the MERS coronavirus, as two candidate viruses, pathogens that worry us as global health threats. And so right now it's investing millions into vaccine candidates to bring them to the point where we've got safety and immunogenicity study in healthy volunteers. And this is something that wouldn't be done because no company wants to make a vaccine for a disease that is not a big public health problem today. So what we need to do is, as a public good, to invest in these sort of public health solutions. So, and not to be miserable, but while Ebola was raging in West Africa, the number of deaths per day from diseases, some of which we have vaccines for, rotavirus, malaria. We don't have a very good vaccine for malaria. We have one vaccine, it's not very good. HIV, we don't have a vaccine for. So whilst the world was focused on quite rightly on Ebola, it was against the backdrop where children were dying in these four countries, with these sort of numbers a day. So clearly there's still some really big goals in the vaccine space that need to be addressed. The last one here is just to talk about the Zika virus. This is Aedes aegypti, the main vector of Zika virus, and you'll know that. And of course, the enormous problem of Zika is not so much the mild illness that you get from getting a Zika infection, but because in pregnant women this can be a congenital infection where the virus infects the fetus and replicates in the fetal brain, and has a range of poor outcomes for infants who are infected in utero. There's an enormous amount of interest in making a Zika vaccine. There wasn't so much commercial interest in making a Ebola vaccine. So what's the difference? The difference is middle-income countries with markets big enough, the vaccine developers can get a return on investment, can make a profit, are being affected by the Zika virus. And so countries like Brazil, the southern United States affected by Zika transmission, Singapore affected by Zika transmission, all of these are candidate markets for a Zika vaccine, and therefore the big vaccine developer companies are interested in the Zika space. And unlike the Ebola example, in the case of the emergence of Zika, this time the World Health Organization gave really strong leadership in helping to frame the strategy to control this illness. And so this is still kind of mostly true. So this is the number of Zika-affected countries in different WHO regions. The epidemic started really the beginning of 2015 in Latin America. There's been over 80 countries affected, including countries on our doorstep in the Western Pacific and in Southeast Asia. The epidemic is waning rapidly through Latin America, so it's very hard to get Zika in Latin America right now. It continues to grumble along in Southeast Asia. So, last slide. So, immune correlates, a really practical, important bit, but a lot of really critical bit of practical vaccine development. Ensures that you can make a vaccine and the same way every time. It's going to work the same way with every batch that you make it. It allows you to understand whether a population's immune or susceptible to a given pathogen. In some settings, it might be able to take the place of doing phase 3 efficacy trials. There's examples of vaccine, for example, against Japanese encephalitis virus, a vaccine licensed solely on the basis of immunogenicity, accepted correlates of immunity data, rather than phase 3 clinical trials. And really important for bridging from first generation to second generation vaccines. I've just told you about CEPI. CEPI fills a gap now in giving pull-through for vaccine candidates against emerging infectious diseases that we worry about. And I've mentioned the MERS coronavirus and funny influenza recombinant viruses that we continue to track in China. And I've told you about the animal rule, told you how that can be used in parallel to clinical safety studies, but how increasingly it's being used to enable fast-tracking of vaccine development. Told you about human challenge studies, a really growing field to enable us to understand disease pathogenesis, but also mostly to try and, most of the time, down-select vaccine candidates so that we don't spend lots of money and time on crappy vaccines that are not going to work in large studies. It lets us throw crappy vaccines out at an earlier time point if they fail in human challenge studies. And of course, again, back to immune correlates, bridging studies are crucial in going from one vaccine to a different type of vaccine. But if you hold the same immune correlate, you might expect to see the same sort of clinical benefits. So I'll leave it there and happy to take any questions. Yeah. Yeah. So most of the time, antibody correlates for vaccines are in fact mechanistic correlates, okay? But sometimes they are not. And so let's take the example of I'll revert to dengue. So we can measure the total antibody response to dengue viruses after immunisation, but they have very little relationship with clinical immunity to those particular dengue viruses. So the total antibody population to a given antigen or pathogen is not necessarily a good correlate of immunity. Yeah. Yeah. Look, so in practice, most of the time we're talking about virus neutralising antibodies, and so that requires a cell culture system, for you to culture your virus and to demonstrate that addition of those antibodies as they exist within a serum or plasma sample from a vaccine recipient, for example, that those antibodies are able to neutralise virus infectivity for a set of cell lines. You could also do passive immunisation studies where you passively transfer a population of antibodies and then challenge them. You don't, if you can't culture your virus, if your virus is difficult to culture or your bacteria is difficult to culture, there's examples of recombinant virus systems that substitute in the antigen of interest, lentivirus systems, for example, where you don't need the pathogen itself, but you've got a sort of substitute viral system to work for you. Yeah. Yeah. Yeah, so here we're looking at ELISA positive responders, and I guess again similar theme. These are people making antibodies that bind to the meningococcal polysaccharide on an ELISA plate, so it's just a biochemical binding, versus antibodies that are functional in that they can bind the polysaccharide, and for meningococcus, the mechanism of antibody function is by opsonizing. So those antibodies bind the polysaccharide and then bind complement in the serum, and then that complement punches holes into the bacterium. And so you can see that, look, I think that if I put confidence intervals on those on these numbers, 75 to 68 is not a drop for this group. So yeah, don't worry about that. The thing